Think Forward. Think Research Channel.
Good afternoon. Thank you, Jim. Thank you, everyone, for that wonderful morning. I'm really inspired and awed, and it's wonderful to hear the work that's being, how carefully the documentation of the accomplishments that you've realized in Botswana is taking place, because I think it really is important to learn from that. And that's the way to do it. This is going to be slightly different. As Jim said, it's focused on a specific issue that has been alluded to in a number of the speakers, and it won't be really so much data presentation, but really a sort of discussion of the issue. And I was asked by Jim to really summarize in some ways the two meetings that were taking place in the last year, both in Boston, one at last year's CFAR meeting, in which there was a portion of the meeting that was devoted to a discussion of TB-HIV interaction, and then, in part on the basis of the success of that, in June, a full-day symposium in Boston, sponsored by the Harvard CFAR, which dealt with a wide and deep array of HIV and TB-related issues, focusing really on the interaction of these two diseases. You all know that this is really a convergence of a perfect storm of disaster, and two major worldwide pandemics are colliding worldwide, more in some areas than in other, and influencing each other in very, very, I think, important and difficult ways. This was the agenda and the issues that were discussed in the June symposium, and as you can see, they were really quite comprehensive. The global picture of, let's see if I can, yes, the global picture of TB, HIV, AIDS co-infection, the need for and new TB diagnostics, particularly in relation to HIV, need for new TB therapeutics in the same way related to HIV, but in and of itself as well. Treating TB and latent TB infection in the HIV/AIDS context, diagnostic difficulties, outcomes or durations of therapy, and treating HIV and AIDS in the TB context, treatment regimens, immune reconstitution, drug interactions, and toxicity. An issue came up which I think had its roots in the initial meeting in November, and actually was brought up first by King Holmes in discussion. And that was something that we're aware of, but I think haven't paid sufficient attention to. And that is the issues of infection control, nosocomial transmission of tuberculosis in the context of integration of TB and HIV, and the expanding rollout of HIV programs around the world. And that was a very important, and I think to this point, neglected discussion. And I was going to talk some about that, but in the interest of time, I think we'll really focus on some of the other issues. And then a discussion of operational issues in TB-HIV co-infection. How do you bring this body of knowledge together in terms of implementation of programs? Dick Chasen and I have talked about this morning, and it doesn't make sense to duplicate a lot of the information, and he's in a much better position than I am to talk about TB diagnostics, therapeutics, and various therapeutic issues in the interaction of tuberculosis and HIV. And I'll present some of the global picture, a little bit about transmission and infection, and operational issues. And I know that everybody's getting very hungry. I wanted to remind us all that this is not a new issue. And many of you remember in the early days of the HIV epidemic here, we experienced a very similar phenomena. And as there was the somewhat naive expectation that in the 80s, TB would essentially disappear in the US, this event occurred. And that is an additional 65 or so thousand new cases of tuberculosis, coincident with the HIV infection, and of course, as we understand now, driven by HIV disease. And that has largely settled down, but was a very important feature of the early HIV epidemic in the US, and now, of course, a major feature of HIV/AIDS worldwide. There are 2 billion people infected with TB, with a wide geographic variation, and about 8 to 9 million new active TB cases a year, the vast majority of which are in developing countries now. Approximately 2 million people die a year from tuberculosis. There are estimated to be 400,000 cases of MDR TB, multiple drug resistance tuberculosis, in a year. And as we know, there are 40 million people or more living with HIV disease or infected with HIV in the world. The intersection of those two epidemics now, estimated, results in about 12 million persons who are co-infected with HIV and TB. Most of the tuberculosis numerically is in Asia and Southeast Asia, particularly India and China, and that would be numbers of cases of TB. And the red represents 1 million or more cases annually. But the TB incidence rate is highest in Africa. And this represents Eastern and Southern Africa with rates of 300 or more per 100,000 per year of tuberculosis. And that, of course, as we understand, is where the most striking relationship between HIV and TB has occurred. The estimated prevalence of HIV and TB cases in Africa and in Southern Africa is greater than 50%. And in the area where I work in South Africa, it's upwards of 80%. So that 80% of new active tuberculosis cases are HIV infected. This has occurred spectacularly and dramatically over time, from 1980 to 2000. This is sub-Saharan Africa. These are case notifications, 100,000 population. And the former Soviet Union, Eastern Europe as well, but not quite as dramatically, whereas the rest of the world has experienced a gratifying gradual decrease in new cases of tuberculosis. Again, this, of course, a function of HIV disease. So to put it in a much more local level, this is a district hospital in South Africa in the years from 1989 to 2000. These are a number of cases. And this is the seroprevalence, antenatal seroprevalence of HIV disease. And as you can see that in these years, as the seroprevalence of HIV has risen in antenatal clinics, up until over a third, actually, at this point in time, so too has the case rate of tuberculosis in that institution. And many of you who work in resource-limited settings or HIV and TB converge will know that in the hospitals, the majority of people now are co-infected. And this is a medical ward in a district hospital in KwaZulu-Natal. It could be the TB ward because almost everybody has HIV and TB. And it does, as you can imagine, create enormous issues that we have not paid enough attention to of infection control and transmission of both drug-sensitive and drug-resistant tuberculosis. So TB and HIV disease are intimately entwined. TB accelerates HIV progression and is the leading cause of death and morbidity in HIV/AIDS worldwide. HIV drives TB incidence and mortality in high prevalence areas, and there's a large and growing proportion of incident TB cases who are co-infected with HIV. Even with successful treatment of TB, those co-infected with HIV often die of AIDS-related complications. And there are major issues in TB which have been previously inadequately addressed or neglected, including diagnosis, treatment, and infection control. And Dick will speak about diagnosis and treatment in a more optimistic way, I think, because this is being corrected. But I think you should know that the way we diagnose tuberculosis in most of the world is the way it was diagnosed 100 years ago, by sputum smear. And there hasn't been a new TB drug in 50 years. And the studies that were originally done that demonstrated TB airborne infection and some of the principles, aerosolation and droplet nuclei, were 50 and 60 years ago, and nothing very much has been done since. So we're dealing with information that is in all of these areas that, without HIV, that is over 50 years old. It's clear that DOTS alone can't control TB in high prevalence areas, and that the HIV response, wherever it is in these areas, has to take into account tuberculosis. So joint TB and HIV collaboration and integration are needed to improve the outcome of both diseases. So the question arises from our operational standpoint, what kind of model of collaboration between HIV and TB programs? And there have been a number of different strategies for doing this in different parts of the world, although it's really just beginning. So for example, in Malawi, I should say, and I think as you all appreciate, in the vertical system of healthcare, TB is treated here, HIV is treated here, and although it's one disease, one patient, the two diseases generally separate the patient into two separate places. What we're familiar with here goes on in the rest of the world, and that creates major problems, of course, in access and I would think in treatment success as well. So one model, which is being done successfully in Malawi, is to take a very successful TB treatment program with established guidelines, endpoints, excellent reporting, and just transport that entire system into the new HIV/AIDS program. There's really no tremendous increase in collaboration. It's really a sharing of previous information. Another model on a much smaller scale, but also very interesting and important, is in Khayelitsha in South Africa, a very famous Médecins Sans Frontières clinic, in which there's been an attempt actually to provide one-stop service for TB and HIV, where patients who are co-infected get all of their diagnosis, treatment, and care in a single facility with the same personnel, quite distinct from Malawi. And there have been attempts at partial integration, some of which I've been involved with in urban and rural centers in South Africa, where some components of each program is integrated and others are remained separate. I'm getting the hook? A little bit. Okay. So I think we could say there's need for many models. One model may not fit all countries. Differences in HIV and TB prevalence, history, resources, culture, expertise, feasibility make it necessary to really think very creatively in different environments. And in fact, one model might not fit even one country because of differences in urban and rural, TB clinic versus primary care clinic, district hospital versus referral center. And it's also very important to exploit existing site infrastructures and not think about this as creating entirely new structures. We tend to think of infrastructure in terms of bricks and mortar, laboratories, even medication, whereas it's the human resources and, in fact, cultural and family strength that is a, I would say, generally untapped resource in resource-limited settings, because HIV and TB actually are not themselves incredibly difficult diseases to treat. We have excellent medicines. We just need to have people take it for the necessary period of time. And that's a human issue, not necessarily a technical issue. So I just wanted to give two illustrations. This is an adherence support group in Tegela Ferry in KwaZulu-Natal, where in traditional Zulu fashion, everybody does their medication construction on a monthly basis with family and friends as a communal exercise. It was very striking and very memorable when these women who were newly HIV infected and started on therapy participated in a community meeting and were to perform. And it would have been the first time that HIV positive women had performed publicly in this community. And they were asked to get up and these women had not previously disclosed their diagnosis publicly. And there was silence, and no one got up. And then after a few seconds, one woman got up, and then another, and then a man, and another, and there were 25 people standing who performed and danced, and it makes me think that although there remain problems with stigma and always will, that treatment always trumps stigma. This is the human resource infrastructure that I think we need to strengthen and rely upon, and it's already there. In many traditional societies, the responsibility of an individual is not just to him or herself, but to the community. And it's not hard to get people to want to serve in the community and serve their family members and neighbors. And this needs really to be, as I said, I think considered as important ways of moving forward as these two diseases are integrated. I think I just want to conclude and have to indulge me. With all the gray hairs I have, I think I can be a little philosophic. So what are the challenges in integrating TB and HIV programs, since I think you'd agree that it makes sense to do it? Well, I mentioned one, vertical systems organized around diagnosis and treatment of individual diseases, where horizontal systems of integration for care for the patient, not individual diseases, is what needs to be aimed at. Requires resources for enhancing and strengthening TB programs, not just HIV programs. And this can, because of HIV epidemic and, as we heard, staff moving from TB programs to HIV programs, risks of potential disruption and overburdening of programs, and a way of dealing that is with integration, not separation. If you use, as I mentioned, existing infrastructure, reducing startup and HIV diagnosis and treatment costs, efficiency may be increased. Is concerned about compromising patient confidentiality, but doing this in an integrated way provides convenient and efficient site for identifying HIV patients. We don't know the optimal timing of institution of HART, and Dick will talk about that, but there are guidelines and we need to learn it, and that shouldn't preclude integration. TB provides only short-term structured treatment support, and HIV treatment is lifelong, but we think it might provide initial structured and supervised HART experience with assistance for transition to self-administration. And there are an array of complexities, clinical complexities, which you'll hear more about, but using an existing structure for promoting HIV adherence, monitoring for side effects and toxicities in terms of the integration of two programs may be actually the way of dealing with this. May result in increased TB transmission, and I think this, as I mentioned at the beginning, a very real and substantial fear and concern that has to be addressed and not ignored, but it also in itself, I think, should not outweigh the the impact positively on TB programs and HIV programs. So I want to conclude with what I think is not a technical, but what is, I think, the most important barrier to integration, and it really is a cultural one, and it's a difference between the cultures of TB and HIV. And this is a major, major barrier. I attended the TB meetings a few weeks ago and found that there were very few people at the meeting who I knew. And I would wager that if TB people came to HIV meetings, it would be the same, and there are probably only a few people in this room who go to both. So they're really very separate worlds. TB culture has always taken a public health approach with firmly established algorithms and standardized measures and outcomes. And it's really been managed mostly through the public sector with minimal community advocacy, if any at all, whereas the HIV culture, as it's grown in the last 2 decades, has had a strong political and human rights orientation. The individual patient, not population-based decisions, generally how treatment decisions are made. There's a lot of familiarity with rapid treatment paradigm changes, turn on a dime rather than move like a battleship. And there's been community advocacy and activism, which has moved the agenda forward. And I'm not saying that one is better than the other because they actually serve different purposes. I think the TB culture needs more flexibility, and the HIV culture needs much more of a public health approach in order for integration to take place successfully. TB services are geared for chronic care of large numbers of patients, simplified regimens, standardization, epidemic control, but a difficulty with individual nuance and new TB diagnostic dilemmas brought on by HIV disease. And HIV services are clinically and patient-oriented with only recent emerging public health practices, very limited experience with TB treatment and public health approach, and I would say, concern and difficulty in actually making that, that turn. In the TB world, HIV has been viewed as an intruder, as it usually is, disrupting TB strategies and programs, but there is an enormous reservoir of wisdom and experience in TB treatment that the HIV world could benefit from enormously. People have described the TB world as being consisted of mature, wise TB men. TB is a challenge for HIV patients, as I mentioned, with discomfort in diagnosis and treatment, and said to be younger, more flexible, and energetic HIV men and women. Well, I'm a mature and wise HIV man, so I think that sort of breaks down a bit. But I think it does sort of capture a little bit of who does the work and the essence of it and where we really need to go with this in terms of integration. I'll conclude by saying that we're really not alone in this, and we're in very, very good company. We can't fight AIDS unless we do much more to fight TB as well. Thanks.
So I'm going to be speaking about the HIV envelope and some recent advances in our structural understanding, and I think the timing of the talks is very nicely done, Jim, in part because we first talked about exit and now we're talking about entry. And some of the differences between the cellular complexities that occur within exit versus the viral complexities, but in a very different way upon entry, I think will become apparent. One thing that I did want to talk about, which is a little bit different from a lot of the other cellular proteins, is that the HIV envelope proteins have a very special function in that in addition to being possible targets of therapy, because they're the primary target of neutralizing antibody, they're also vaccine targets. And I just want to focus a little bit on that because, as everyone here knows, structure-based drug design has revolutionized HIV therapies and led to the development of the HIV protease inhibitors. Why hasn't the same types of paradigms been applied to vaccine design? And so that's a question that seems very reasonable, and I sort of wanted to just spend the first 3 slides and go through that before I go on to some of the other structural work. So in general, if you talk about structure-based drug design, and I'll just give one example here, one normally starts with a lead. In this case, this is actually a little mimetic of GP1 of CD4, and one has a target that one's trying to interfere with. In this particular case that we're working with in collaboration with Claudio Vita, we're trying to interfere with the interaction between GP120, which was the target, and its interaction with CD4. And the lead that Claudio came up with was CD4M33, which is a little scorpion toxin that he had transplanted the GP120 binding surface of CD4 into the toxin. We were able to go crystals of that, and you're able to get that complex. And through current collaborative iterative optimization, we're trying to optimize this complex. And we can do so because we can look directly at this complex and use the atomic level information from that to do this optimization. So what happens in structure-based vaccine design? Well, if you take a direct analogy of what we did previously in structure-based drug design, the lead in this case would be one of the envelope proteins, for example, GP120. But in this case, the target is the entire human immune response. And we've tried to get crystals of that, but it doesn't work. And so you don't get the complex, and so iterative optimization is not possible. Is there hope? Well, there is some hope because there's other ways to perhaps provide surrogates for the target. And one thing that might be possible, for example, if one wanted to elicit a CD4-like antibody, would be to start with a lead, for example, a conformationally stabilized GP120. And there the target is not the entire immune system, but perhaps an immune mimic of CD4. One can make crystals, of course, with GP120 and CD4, but in this case, CD4 is used as a surrogate for the immune response. So one can take indirect means for vaccine development. The iterative optimization then is possible, but the optimization is indirect. So the extra little step that goes in here is animal studies, which tell whether or not these conformally stabilized pieces are actually eliciting the type of antibodies that we have. But that's something that's separate from this direct closed pathway that otherwise occurs within drug design. So this is sort of a schematic of HIV entry. And the one real nice thing that Wes went before me is you can see the dramatic differences in the number of factors that are involved in viral entry versus viral exit. First of all, there's only two proteins. It starts off as one protein, the 160, which is cleaved into the 120, 140. That binds to the CD4. Large conformational changes occur, which then allow binding to the co-receptor, and that triggers entry with the fusion of the virus up here with the target cell membrane. Very, very straightforward, simple process, of which we now have some structural information. Earlier this year, Bing Chen and colleagues in Steve Harrison's lab published a structure of the unligated SIV-GP120, which is a picture. Is that like focused? I don't know if you can focus that a bit. In any case, there's this structure, and Gary Nabel spoke a little bit about that earlier, about how we can see that it's highly glycosylated. There's this outer domain, and how conformational changes can occur upon binding to CD4, which is the original structure that I published with Rich Wyatt when I was working with Wayne Hendrickson and Joe Sadrowski several years ago with the CD4-bound GP120 structure. So we've recently also obtained the structure of the entire co-receptor binding surface. So that is GP120 bound to CD4. We have an antibody in there called X5, but we've got the entire co-receptor binding surface, which is the region of V3, as well as this bit on the core called the bridging sheet. We know from the V3 sequence and its structure, here's its structure here, and its sequence, quite a bit about its information. First of all, we know that its base had extended off of the core, and we could see atomic level details of that. There's a flexible stem as well as a tip that's relatively highly conserved. And here's some atomic level details. The stem had some really interesting things that weren't predicted entirely from the sequence analysis. One of them is that there was a conserved arginine and a proline, which caused a spraying out, a separation of the stems. And so you had this V3 region that wasn't just this loop extending off, but actually was forced to spread apart, and that enhances its reactivity. There's also the part on its tip that is thought to interact with co-receptor directly, and we could see that there's a conserved structure there that was seen in lots of other antibody complexes with peptide that Ian Wilson's group had determined. One thing that we did is we took the structure that we had of GP120 with the V3, and we modeled it into a trimeric structure that we had obtained by optimization of quantifiable surface parameters. This actual structure was produced in part by a nice suggestion that Wes gave us when he was reviewing this paper, and he came back with this nice suggestion that we should do something. And I didn't know who gave that review, but I said, yeah, this reviewer gave this fantastic suggestion. And Wes said, oh, that was me. So sometimes reviewers are very good. Anyway, so we got this structure, and that gave us an idea of how to place our structure within the context of entry. That is, here we have the host membrane, and here's the virus. And we can see very clearly here that the V3 is just sticking right out, right below this entire core to engage prominently with the co-receptor, which would be down here. And that led to this model where you've got the GP120, the V3 stands out with its flexible legs, and then the tip here, which is highly conserved, would interact with the co-receptor tip. So what I'm going to launch into is something that I prepared last night because I've been doing a collaboration with Jim looking at actually how V3 functions. And I wanted to sort of, since he invited me, talk about something that he was interested in. So I'll start with this idea and structure analysis of CD4I antibodies, and in particular how antibodies mimic CCR5. So just some background, the entry of viruses I've been telling you is mediated by sequential binding of CD4 first to create this bridging sheet that then allows binding to the co-receptor. CCR5, which is shown here, has a highly sulfated, has a tyrosine sulfated N-terminus. It's very negatively charged, and there's a bunch of tyrosines which have a very special modification of being sulfated. This region here appears to be mimicked by CD4I antibodies that bind to the same region on GP120 as the co-receptor N-terminus. So we're interested in this for a number of reasons. One of them is that through work with George Shaw, we showed that HIV-2 could be neutralized by HIV-1 when it was triggered by CD4. And a lot of other work showed that this was related to the CD4I antibodies. So if one took this infected individual, patient 133M, and looked at its HIV-1 virus, its viral titers against its own virus sort of changed over a period of time, 2 years there. But if you looked at the neutralization of this individual's sera against an HIV-2 virus, there's absolutely no neutralization. But if one first triggered with CD4, one found that one had increasing levels of neutralization. And what George was able to determine was all this neutralization was due to antibodies that recognized the co-receptor binding surface. And so what that meant was that one could have, through the difference between HIV-1 and HIV-2 evolutionarily is enormous. There's enormous evolutionary distance. Yet one could have antigenic recognition and neutralization if you had exposure to this co-receptor binding surface. So that was, I think, really quite striking, and George published that, and I think I sort of summarized that. This extraordinary breadth of neutralization attests to the conservation of the co-receptor binding surface all the way from HIV-1 to HIV-2. So a number of years ago, we were interested in these CD4I antibodies, and we obtained a number of them from Mikko Dimitrov and James Robinson, and started sequencing them. And so we obtained a lot of sequences of them, and there wasn't anything that really stood out, except for some of them were very, very long and had a lot of negative charge and a lot of tyrosines. And why is that interesting? Well, it's interesting because the N-terminus of CCR5, as I told you, is negatively charged and has a special tyrosine modification, which is the sulfation. The signal for tyrosine sulfation is that it's just negative charges and has tyrosines there. And one can run these things through programs and see that a number of these that are longer and negatively charged are actually should be tyrosine sulfated. Since we had sequenced these, we actually had all the DNA, cDNAs. We sent these to Mike Farzan, and Hoyoung Cho did this with Joe Sadrowski, and actually looked specifically to see if tyrosine sulfation was actually occurring on the antibodies. That is, the antibodies are binding to the same site as CCR5 that's sulfated. Do you actually get the same type of post-translational mimicry on the antibody that you could see on CCR5 itself? And the answer is that you do. This is one experiment here with 412D where you can see that it's sulfated, but if one changes the tyrosines to phenylalanines, one no longer gets binding at all to ADA virus. You get a teeny bit of binding to YU2 and also to GRFL, but basically this 412D was a functional mimic of CCR5. So we analyzed a bunch of antibodies in this manner and came up with the following, that there were some that were short in sequence for the CDRH3 regions, but a whole bunch that were long, and some were long and functionally sulfated, and in that manner replicated, mimicked in some way, the N-terminus of CCR5. In order to analyze these things further, we obtained crystals of CD4I antibodies. These are the uncomplex crystals, and these crystals are shown here. And Chi-Ching Huang and my group solved those structures. And the bottom line is that for those things that we weren't that interested in, that is the ones that weren't sulfated, we could see quite well. But the ones that were all sulfated functionally, all the CDRH3 loops, which were long, unfortunately were almost all disordered. So we weren't able, unfortunately, to see the most interesting part of this molecule. So what do we do next after that? Well, it turns out that the story gets a little bit more interesting because Xihuo Zhang and Joe Sadrowski did a really interesting experiment with Mike Farzan. And what they did is, in order to really explore the functional mimicry of this antibody 412D and CCR5, they decided to take the C05 N-terminus, which is here, and this in yellow is shown the 412D C0H3 loop. And they actually did a swap of them. So they made the N-terminus of C05 a chimer with the CDRH3 loop of C05. And that actually gave functional entry. So C05 is shown in this sort of light blue, and then the purple is shown for the chimer. And you can see for a bunch of viruses that entered fine. So the CDRH3 loop that is sulfated of 412D is a functional mimic and makes a good chimer, replicates the ability of C05. And so we thought that was quite extraordinary, the ability of the immune system to really mimic to this precise degree C05. So now we're getting to stuff that I thought Jim would be interested in, which is the structural determination of the YU2 core plus V3 in combination with 412D FAB. And so Chi-Ching was able to grow crystals of this complex. This is the core with the V3 added on from the YU2 strain. It has CD4 in it, 2 domains, and these 412D FAB antibody that's the mimic of CCR5. And she solved those, collected data about 3 weeks ago. She was able to solve that, and this is the initial data shown here on the right, and we can see the V3 loop fitting into this data. Okay, these are the initial maps, and usually the initial maps of molecular placement aren't so great, but upon refinement, this will look really quite good. One of the things that's quite interesting is, of course, what does that CDRH3 look like that should be sulfated? And so here's the initial map of the CDRH3, and there's these 2 blobs sticking out here and here. And so one could trace those, and you could see these 2 blobs still sticking out there. And those turn out to be the positions of the 2 sulfates. So it's a slightly different orientation, but if you put in the sequence as tyrosines, but don't put in, you see these large positive blobs in the FO minus FC map at 3 sigma sticking directly over where the tyrosines would be. So that's, of course, where the sulfated tyrosines are. And so now we have a real atomic level picture of GP120 in its CD4 bound conformation. We've got this little V3 loop out here, and we've got the functionally sulfated tyrosines actually in here as well. So just to summarize some new structures, these are of CD4, GP120, 2 V3 loops. You can see that the core parts are very, very similar. There's a flexible stem that can change a little bit, and then the base is very similar, both forms of beta turn with a beta hairpin turn with 2 different antibodies. This one is the 412D, which is a functional mimic of CCR5, and the other one's an X5 antibody, which is quite broadly neutralizing on its own. So here's a summary and future directions. The structures of the unligand and CD4 bound forms of GP120, we now have these, including multiple views of the core and the V3. There's still many important steps in the HIV entry pathway that remain to be characterized. We feel that these investigations in the CD4I antibodies really reveals the remarkable mimicry and the ability of the immune system to mimic in a precise way the post-translational tyrosine sulfation of the N-terminus of CCR5. Structure of 412D actually shows this atomic level mimicry shows this mimicry in atomic level details. We're currently attempting to visualize the structure of CCR5 in its interaction with GP120. There's 2 questions that we're quite interested in. One is, what is the degree of antibody mimicry? To what degree does that actual CCR5 look like the peptide 412D that we have binding, the antibody 412D that we have binding? And then, can these structures be used for drug design? You can see the remarkable conservation between HIV-1 and HIV-2, where antibodies can actually bind that surface and neutralize. If one had a small drug that bound to that surface, and one can see that one can make that from the atomic level information that we're now providing, perhaps that would be a good entry inhibitor. So the primary, I've mentioned a number of names here. We collaborated a lot with Mike Farza and Joe Strzalski. People provided reagents like Mikko Dimitrov and Yang Zhang. Crystallization attempts were really aided in this by Robin Stanfield and Ian Wilson, who turned me on to the idea of robots. I initially resisted and then realized that these things are very useful because they stop you from doing all your work and spending your time there and just do all your crystallizations for you. James Robinson provided a lot of the antibodies, including the 412D and the other functional sulfate antibodies. Rich Wyatt and Ming Tan provided proteins, and Qi Huang in my group was really instrumental in solving all these structures. So she's just been phenomenal. So I'm welcome to take questions, and I think I'm under my time limit.